Hi, I'm Lauren Volpert, and this summer I spent 1 month at the Weill Cornell Medical College working under Dr. Mayad in the Department of Cellular Development and Biology. This was my second summer working at the Mayad lab. We study salivary gland morphogenesis and the effect of Sark gene overexpression in the Drosophila melanogaster. Drosophila melanogaster, the common fruit fly, is a model organism used for research in development and molecular biology. Because of its small size and short generational time, Drosophila is used for all types of research, and results are often applicable to humans. Drosophila are one of the few species of which the complete genome is known. We study the salivary gland morphogenesis in Drosophila because it can give us insight into how organs and glands are formed. Since this was my second year at the lab, I was able to move forward with my research and begin pilot work on a new lab grant at the Mayad lab. Last year, we researched how various genes affect organ development by crossing different genotypes and looking at the resulting embryos. We know the genes that are essential for morphogenesis, specifically in the salivary gland, in cell culture. However, the mechanism by which these genes control the coordinated movement of cells in a developing tissue is largely unknown. The salivary gland undergoes a process called invagination during embryonic development, during which the salivary gland migrates and turns. The salivary gland is a useful phenotype to study because it is visually distinct during embryogenesis, and therefore the effects of gene deficiencies are easy to trace. We then take microscopic images of the embryos and look at the gland morphogenesis. In this set of 4 pictures, you can see the process of regular invagination from stages 10 to 14 of embryonic development. We look at the germ band, which retracts itself throughout embryogenesis to identify the stage. The blue arrows indicate the start of the germ band in each stage of development. The new grant enabled the lab to research a common cancer gene called Sark, short for sarcoma, which means malignant tumor, and how it reacts to being overexpressed in the salivary gland. In order to create this exact combination, we crossed the UAS Sark 64B genotype with the FKH-GAL4 genotype. As you can see from this slide, the names of the genomes correspond to the factors that cause the metastasis. UAS, the upstream activating sequence, or what turns on the gene, activates the Sark gene, which causes metastasis, or the production of the sarcoma. But the FKH is what promotes expression only in the salivary gland. One of the most interesting parts of being at the lab was learning just how much we are able to manipulate genotypes in order to achieve a desirable result. When these 2 genotypes are crossed, for example, they are able to promote overexpression of the Sark gene only in a specific area of the embryo, the salivary gland. We then looked for the resulting mutations and began to categorize them. So the big question I always get asked when explaining my research is, what are your results? This was a pilot study, and the research is ongoing without definitive results at this time. Our lab is working to find the exact genes that enable invagination and Sark overexpression, and we have been able to isolate a small region where these genes are thought to be. The lab also hopes to more fully understand the process of metastasis, and in being able to replicate it, there is hope of being able to stop it. I would like to thank Dr. Mayad, Unisha Patel, and everyone else at the Mayad lab for everything they taught me during the past 2 summers. I would also like to acknowledge Dr. Cox and Ms. Finlay for all of their help. Thank you.